Assalamu alaikum. This is Tehneem Fatma and I am doing MS Chemistry from UMT Sialkot. I have written a research paper on beta lanes. Now, let's see table of content. Table of content includes abstract, what is beta lane, classification, structure, occurrence of beta lanes, role of beta lanes in plants, beta lane extraction, beta lane biosynthesis, application of beta lane as a natural dye in food industry, in cosmetics, and in textile industry. Conclusion and references. Now, abstract. We have discussed the betalain in this paper. Betalain are the class of compounds that are found in Centrospermi family and are classified into two main classes. The structure of betalain consists of a base which is beta-lamic acid. We have discussed the role of betalain in plants and their biosynthesis and also how they extracted from different plants. At the end of the paper, we will discuss the application of betalain in different industries. Betalain used as a natural dye and it shows different behavior at different pH. Betalain has antioxidant, anti-inflammatory and antioxidant properties. Color derived from the plants are safe and environment friendly. Now, what is betalain? Betalain is a class of plant pigment exclusively found in Centrospermi family. Betalains are nitrogenous, water-soluble, red, violet, and yellow pigments that are found one of the major pigment classes providing striking color to the plant organs alongside chlorophyll, anthocyanin, and other flavonoids and carotenoids. Due to their strong antioxidant properties, they have the potential health-promoting properties which includes anti-cancer, anti-inflammatory, and anti-diabetic activities. Also, their solubility in a wide range of pH has made them a pigment of choice for food industry, where they are widely used as a natural colorant. Now, what is the classification of betalain? Betalain are classified into two subclasses, which is beta-xanthine and beta-cyanine. Beta-cyanine are red-violet in color, while beta-xanthine are yellow in color. Beta-cyanine are further classified into four subclasses. Now we will discuss the structure of betalain. All the betalain are based on a common beta-lamic acid, which on condensation with cyclodopa derivative are with various amino acid and other amines to form a beta-cyanine or beta-xanthine. The condensation of a beta-lamic acid with various compound determine the different differential appearance of two betalain groups. Beta-cyanine show maximum absorption peak at 535 to 538 nanometer, while beta-xanthine typically show highest absorption at 460 to 480 nanometer range, depending upon the molecular structure and type of solvent. Till now, up to 75 betalain has derived from 17 different plant families. Now, we will discuss the occurrence of betalain. About 17 cyclophyllae. After this, we will discuss the occurrence of betalains. About 17 cryophylales families contain this pigment called betalain. Observingly, anthocyanin and betalains have identical functions and have never been found together in the same plant. So, apparently, they are mutually exclusive. Betalain are hydrophilic in nature and are accumulated in the vacuoles of the cell, mainly in the epidermal and subepidermal tissues of the plant, which synthesize these pigments. Betalain give color to the flowers of many genera of plants such as Mirabilis, Gluteophyllum, etc. In cryophylales, anthocyanin determine the phenomena of coloration in the family of Cryophyllaceae. Recently, a natural occurrence of betalain in a bacterium family has been described. The bacteria Gluconobacter diazotrophaceus were reported to produce dopaxanthine. In the figure 1, we have the structure of beta-xanthine and beta-cyanine. The structure A is the structure of beta-cyanine, in which R1 may be hydrogen or sugar motifs, while in the structure B, R4 usually be hydrogen and R3 can be amines and amino acid groups. And this is the structure of beta-lamic acid. Role of betalain in plants. Betalain occur in a wide range of plant tissues, including leaves, stem, fruits, flowers, roots, and seeds. Like other plant pigments in the flowers and fruits, betalain plays an important role in the attraction of pollinator and also for the fertilization and for seed dispersal. Second one is betalain plays an important role in the defense against biotic and abiotic stresses, and they play an important role as compared to the anthocyanin counterpart. Betalain in the plant photoprotection. Betalain in the plant photoprotection showing reduced damage to the photosynthetic capacity in the red pigmented versus green pigmented leaves. Betalain production is upgraded when the plants are exposed to the light or to the UV radiation. Similarly, the increased betalain accumulation under the drought and the salt condition indicates a role in the protection against these stress factors. Next one is protection from abiotic stresses. The protection from abiotic stresses coincide with their occurrence in cryophylls. Most prominent feature is its their dominance in the arid and semi-arid region and habitation of saline and alkaline soil. The member of several families are particularly adapted to the arid and saline region. For example, ice plant family, porcelain family, and cacti family. Betalain shows strong antioxidant properties, as in case for anthocyanin. Next one, involvement of betalain in the plant protection against biotic stresses. Plant protect Plant protect against biotic stresses is less characterized, and the role of betalain in defense against pathogenic fungi has major factor driving their evolution. And the last one is radical savage activity. Radical savage activity causes the delayed death and proliferation in the necrotrophic fungus. These are some plants which contain betalains. Now, we will discuss betalain extraction. The material which contain betalain can be macerated or ground. Pigments can be water extracted, and these can be extracted by using methanol or ethanol solution of 20 to 50%, and this is required for the complete extraction. However, the aqueous extraction promoted the better stability of the pigment, while the slight change in the pH of the extraction medium enhances the beta cyanine stability and also avoid the oxidation by polyphenol oxidases. Enzyme pretreatment is very important, and the heat treatment of the exact extract is desirable, and this avoid the betalain enzymatic degradation. Enzymatic treatment for the degradation of hydrocolloids may also favor to the pigment extract. Now, fermentation of the extract reduced free sugar by increasing the beta cyanine contact. Cell permeability enhances betalain extraction efficiency with relatively low levels of tissue damage and low energy consumption, and this effect was attributed to the cell permeabilization. While on the lab scale, betalain may be extracted by using different methods such as diffusion extraction, solid liquid extraction, and reverse osmosis, and also by ultrafiltration. And these processes are more efficient on the recovery of betalains from beetroot tissues. 80% of the beetroot juice solids contain carbohydrate and nitrogenous compounds. Now, in this picture, we will see the bioactivities of betalain that are anti-inflammatory activity, antioxidant, immunomodulatory activity, and cancer properties. From the red beet, we extract the betalains. Betalain can be beta-cyanine and beta-xanthine. Novel extraction technologies contains ultrasound, microwave, pulsed electric field, and membrane technology. Now we will discuss the betalain biosynthesis. Betalain are synthesized from triosine. Triosine is an aromatic amino acid that is mainly produced in the plant via Shikimate pathway. Triosine is initially hydroxylated to form 3,4-dihydroxyalphenylamine L-DOPA is subsequently converted to beta-lamic acid, and beta-lamic acid is the core backbone of all the beta-lamic compounds. This is the two-step reaction. In this reaction, enzyme used is 4,5 dihydroxygenase enzyme. After this, alternatively, L-DOPA is oxidized or cyclized to form cyclodopa, which spontaneously condense with beta-lamic acid to form beta-cyanine. Betanidine is next react with the sugar at 5O or 6O position to form betanine or gomphrene, respectively. A variation of the core pathway towards the betanine formation occur in the same cryophylales families. In this, cyclodopa in the first step react with the sugar at 5O position and then by the condensation form cyclodopa 5O glycoside with beta-lamic acid and directly produce betanine, while the formation of yellow xanthine occur via spontaneous condensation of beta-lamic acid with amino acid rather than first it is converted into cyclodopa or into cyclodopa derivatives. Betalains are though can be synthesized in the cytoplasm or endoplasmic reticulum based on the subcellular localization of their key biosynthetic and the secondary metabolites eventually stored in the vacuoles of glycosides. Now, we will discuss the application of betalain as a natural dye. In the food industry, betalain have been used as a natural colorant since the turn of 20th century. The early application involved the use of pokeberry juice to improve the color of red wine, and it was forbidden in France in 1892 because of the juice also contained purgatory saponins in it. The beetroot red extracted from red beet is commercialized by the European Union and USA as a food colorant. In Betanin from red beet is one of the successful food colorant which is used in food industry and is approved by US FDA and European Union. Red beet colorant can be effectively used to color the dairy products such as yogurt, ice cream, hard candies, fruit juice, frosting, cake mixers, gelatin, poultry meat, etc. Now, beetroot are notorious accumulator of nitrates and nitrites during growing and there is necessary to reduce these level in the red beets. In particular, the yellow orange prickly pear fruit are the promising potential because of the yellow water soluble pigment. The low level of colorless phenolic compound in the prickly pear is due to the beta lanes with these phenolic are avoided. Prickly pear contain high amount of reducing sugar and amino acid and thereby may enhance Maillard product formation. Next, concentrated beta-cyanine extract from the fruit of ostraca. Red purple powder from the ostraca fruit juice was successfully applied as a natural as a food colorant in two type of model systems. One is in the yogurt and other one is in the soft drinks. On the other hand, amaranthus plant, which can be grown in the wider range of environment and have the potential source of 49 betalains. Beta-cyanine from amaranthus plant shows high colorant, high potential and used as a natural colorant in jelly, ice cream and beverages. The red pitaya. Now we will discuss the application of betalain in cosmetics. Additionally, the same coloring substances may also be used in the pharmaceutical and cosmetics. Lipstick was created with betalain pigment, which were extracted from dragon fruit as an alternative to synthetic color, and it showed the minimum side effects and allergic or it never dried the lips. 4% of dragon extract was added in the lipstick formula. Lipstick formulated with betalain pigment extracted from dragon fruit displayed satisfying level of coverage, spreadability, smoothness, and stability when it was stored at the room temperature. While the amaranthus juice has also been used by the by women as a facial cosmetic. To improve the stabilization of these coloring agents, we use ascorbic acid, isoscorbic acid, and gluconic acid, etc. In textile industry, normally the red extract obtained from red beet peel does not generate any color to the wool. And the dyes extracted from the both pulp and beetroot peel have no affinity for the textile. Because in the wool there is no affinity between the two parts in the dye bath. Wool also contain the same type of functional and non-ionized groups that are COOH and NH2 groups. The ionization of these groups to COO negative and NH3 positive occur only during deprotonation and activation process. Thus, the attraction between these ionized groups with the formation of electrovalent bond and generation of the red coloration to the wool. The main constituent of wool fiber is keratin. Keratin is a fibrous protein whose polypeptide chain is comprised of 18 alpha amino acids, which are well established in a relative ratio and in sequence. It shows amphoteric behavior in aqueous solution. At the pH value lower than isoelectric point and the positively charged protonated amino acid can interact with anionic species, which were generated by acidic dye, while at the pH value higher than the isoelectric point, the negatively charged deprotonated carboxyl group can interact with cationic species as a cationic dye. The wool dyeing with cationic dye is seldom. Usually, synthetic and natural acid dyes are used for the coloration of wool. Now, extraction of dyes. Extraction of dyes from the pulp of beetroot from the for food industry can be done in the presence of water or in the acidic or alcoholic medium. After this, stability of betalain. The stability of betalain is influenced by various factors. For example, pH, temperature, dyeing content, acylation degree, antioxidant, chelating agent, etc. By modifying these factors, the color of dye can often be affected. The protein material, wool or silk, are not dyeing in the fresh beetroot juice extract. If the mordant is added to the dye bath, then only light color such as milk rose color or pink baby color can be obtained. The silk dyeing using simultaneously a moderating technique in the presence of eco-friendly acid moderant, citric acid and tartaric acid led to the good colorant, fatness and the property and some changes shed to the depending upon the moderant. Now, color fatness, color fastness properties are very good in two cases. First one is when wool has been dyed with an extract functionalized in acidic environment, or the second one is when the wool has been functionalized in an acidic environment before dyeing. This is important because we have discussed previously that wool have the same functional group as dye also have the same functional groups. So functionalization of these groups is very important. For this we use functionalizing agents. In this picture we will see that beetroot peel, we added the beetroot peel and the water for the extraction of beetroot extract, then filter the extract and add this extract to the dyeing and wool, and color fastness properties are very good in two cases. First one is that when wool has been dyed with the extract functionalized in acidic environment, and the second one is when wool has been functionalized in acidic environment before dyeing. In this picture we will see that we have the extract and functionalizing agent. First we add functionalizing agent to the wool and wool is functionalized, and then we add this functionalized wool into the dye for dyeing. And the second method is that in which first we functionalized the extract, betalain extract, and then we add betalain extract to the wool for dyeing. This this acidic environment is responsible for the formation of betalain deprotonation and wool activation. The pH value influences both the content of betalain and the color characteristics of dyeing wool. At the end, within 10 years, accordingly, about 143 papers have been published in which the word betalain appears as a title of an article. When the research is extended to the article title, abstract, and the keyword, result shows that 634 papers have been published. Additionally, when the research is extended with the word colorant, dye, or pigment, there are 95, 82, or 362 papers published respectively in May 2021. Conclusion. Betalain are the food color. Betalain are natural colorant. Betalain are present in the plants such as beetroot, Swiss chard, amaranthus, cactus pear, red pitaya, and some tubers. Betalain extraction can be done in the lab by diffusion extraction, solid extraction technique, and osmosis. Betalain can be biosynthesized from triazine, though shikimate pathway. Betalain have no harmful impact on our health and have antioxidant, anti-cancer, and anti-inflammatory property. And it also used in cosmetic industry such as in lipsticks and also used in facial. In textile industry, it was used to dye wool, silk under specific condition. And the plant driven colors are safe and environment friendly. That's all from my side. Thank you. Allah Hafiz.